If not, whatever. There we go. All right. Well, thanks for joining.
Yeah, got a lot of material split between today and tomorrow. And I'm looking forward to it. I hope you don't mind if I just kind of go through my presentation. It's got a pretty tight logic. So typically when I do one of these presentations, I don't do it kind of Q&A. I save Q&A till the end. Is that okay?
Yeah, that's what we did with Casey. So why don't you start and let's get going.
Great. Can you enable screen sharing for me?
Sure. Let's see. I'm just going to make you host. How's that?
All right, awesome. Let me do this. And scroll back up here. Whoops. One sec. How does that look?
That looks good to me.
Good. So my name is Paul Nelson. I'm a senior fellow at the Discovery Institute. I've been there since 1996. And I work professionally on this question of the testability of the theory of universal common descent. So in this presentation, which is divided into two halves, I'll do the first half today, the second half tomorrow. I'm going to ask the question, can universal common descent be tested by molecular data? And I want to begin with laying out the historical and logical framework within which this question takes its significance. And of course, for any biologist today, that is a Darwinian framework. In particular, in The Origin, very close to the end of the book, Darwin argues the following: the affinities of all the beings of the same class have sometimes been represented by a great tree. I believe this simile largely speaks the truth. All living things have much in common. So he's going to extend his argument from the great classes of plants and animals to all life on Earth. All living things have much in common. Therefore, I should infer from analogy that probably all the organic beings which have ever lived on this earth have descended from some one primordial form into which life was first breathed. It's interesting to note, actually, that in the 19th century, Darwin was very much the exception among evolutionary thinkers for arguing this way, namely that there was one great tree of life. His contemporaries, such as Ernst Haeckel, did not think that that was the case, even though Haeckel, produced some very famous branching diagrams, trees of life. Haeckel actually thought that spontaneous generation was occurring all the time, so that the Monera, the single-celled organisms that he observed, were not the lineal descendants of cells on the early Earth. So Darwin, I think you can make a strong case that Darwin really is the person who put this theory of universal common descent on the map scientifically in a way that, as I've said, we still inhabit that scientific universe. And you can trace conceptual linkages from the origin right to the present. So affinities today we understand mainly in terms of molecular data, which is what I'll be talking about today and tomorrow. A great tree is this theory of universal common descent. And some one primordial form is now referred to as the last universal common ancestor, typically abbreviated LUCA. So here is a representation, a sort of textbook representation of what one would be likely to see. I guess I can't really move that. I don't know if the viewers can see that. We'll just have to live with it there on the screen. The screen.
It should look fine, but I think they.
All right, most of the diagrams will be visible. So this doesn't look like a tree. This is the work of Norm Pace at Indiana University. But we can imagine it as a tree if we imagine that we're hovering over it in a helicopter, let's say. So this would be a great oak tree with three major branches, which are known today as the domains of life. So we have the bacteria, single-celled organisms lacking nuclei, the archaea, a different group of single-celled organisms also lacking nuclei, and then the eukaryotes here. These are organisms with nuclei. Of course, we're on this branch here, the animals. And the last universal common ancestor is here. It's not at the origin of life. Standard theory tells us that the last universal common ancestor actually occurred as a cell at some point after the origin of life itself. But the logical thing to bear in mind with this sort of figure is its universality. That is, there's nothing outside the domain of this theory. It's universal with respect to life on planet Earth. And that universality is very attractive to biologists, as I'll be discussing in more detail in just a moment. One can think of biology as a science of exceptions, where for every generalization that one forms, you can find a species that does it some other way. But in this case, this is a proposition that extends across the whole of terrestrial biology. And for that reason, it's very valuable to biologists. And for that, I think for that and other reasons, they're loath to give it up. Now, this might not be true, right? Can it be tested? Well, the short answer, counterintuitively, is no. But also yes. Now, someone might say, well, Paul, that's a contradiction. You need to go back and retake Logic 101. In fact, explaining why this contradiction exists within evolutionary theory today and why it matters will be the point of my presentation today and tomorrow. So we say, well, it's not testable, but it is testable. Well, how can that be the case? Well, I'm going to show you. So we'll take this universal common descent topology as the canonical history for life on Earth. But as I'll explain in much more detail tomorrow, this geometry is now fracturing at its base, largely as a consequence of genomic data that no one could have anticipated throughout really the whole of the 20th century. In fact, I was nearly done with my PhD in the mid-90s when whole genome DNA sequencing became a practical technology. And that technology has radically shaken or undercut certainty about the truth of universal common descent in a way, again, that most evolutionary biologists, especially within the neo-Darwinian framework, they would have found that hard to believe. So to give you the punchline that we'll come to tomorrow, the domain of universal common descent is fracturing. The tree, to use a different metaphor, the tree is coming apart at its base. And the significance of this going into the 21st century for biology can't be underestimated. So here's a little outline. We'll first define the theory. And again, this is very important because it's only when the theory is carefully defined that we understand the significance of the data I'll be discussing, especially tomorrow, for the challenges that the data raise. We'll ask, how does this theory generate predictions? And then we'll give the no answer. We'll say, this theory cannot be tested, and I'll explain why. Then tomorrow, first thing off the bat in the presentation tomorrow morning, I'll give the yes answer. And then we'll conclude with what I think is the heart of the problem, something that I'm calling Sober's paradox from the work of University of Wisconsin philosopher of science Elliot Sober, a fascinating puzzle that really goes to the heart of how not just molecular data, but comparative data generally raise problems for evolutionary inference. So today, logical historical analysis, begin with some molecular case studies, and then tomorrow, molecular case studies in more detail. So this is what we'll focus on today. And again, as this slide indicates, the logic counts because that's what gives the data their full significance. All right, defined. Now, it's surprisingly widespread in the biological literature to regard universal common descent as a deduction from a pair of self-evident biological premises. And I won't spend too much time on this, but I do think it's worth talking about. And the reason is one should ask oneself, why do biologists frame it that way? Why do they regard it, many of them, as a deduction from these two apparently self-evident premises? So keep that question in mind. So Keith Stewart Thompson spent much of his career at Yale. I believe he's now in the UK working at Oxford. But in a paper in the early 80s, he said, universal common descent is a deduction, and it comes from the premises, one, that life arose only once on Earth, and that all life proceeds from existing life. He says, given these premises, and if the pattern of diversity among organisms has changed over time, then it must follow, deductively, that any new forms have appeared as the result of a process of descent from pre-existing forms. Universal common descent is based upon such unassailable logic that we act as though it is a fact. Okay. Thompson was hardly alone in this opinion. So in 1994, in a letter to me, Richard Lewontin, who died last year, actually, I think it was on July 4th, a wonderful biologist, kind of a hero of mine in many ways. In a letter to me in June 1994, he put it this way: let me sum up the materialist position on evolution. We take it as given that living material arose from non-living only once in the very distant past. And I'll abbreviate, I won't read the whole passage here. He says, these things, these entities, living things, had to be simple. They couldn't look like an animal, a metazoan, or a vascular plant. Two, now you can see he's building a deduction here. Since that time, there's been no more events of abiogenesis. It happened only once. That was a unique event. And so we have the law of all life from life. It's known as the law of biogenesis. Three, there are organisms alive today which were not here 2 billion years ago. So it's again the same argument that Thompson is making. He says, if you take these things, these premises as given, evolution follows as night to day. It has to be a continuous chain of life from the very first cells. That is what we mean by evolution. All the rest is commentary. And then he adds with great candor, there's no way we can prove any of these things. And in fact, as I said, many biologists, surprisingly many, regard universal common descent as an axiom or first principle of biology. So Kenneth Weiss at Penn State, for instance, in an article in 2001 with an interesting title, "We Hold These Truths to Be Self-Evident," he said, among the basic principles of Darwinian biology, and by the way, this is not a typo, for Weiss, Darwinian theory is so deeply embedded in the structure of biology that it can be treated as a generic adjective. So that's not a typo. That's a deliberate choice in his writing. All life comes, or excuse me, all life came from a point source. So you have this abiogenesis singularity at the beginning. He says the assumption of a point source is important to the entire theory. We insist upon spontaneous generation in the beginning, but then forbid it. What beyond shared ancestry is inviolate? So, sorry about that. So you can do this. And actually, it's a rather elegant argument. Infer a single event of abiogenesis. There it is. Never mind how that happened. Just say there was a cell. That process, whatever it happened to be, then ceases. You have a single cell present there. No more abiogenesis can happen. Well, if that is the case, then everything else on the planet must trace its ancestry to that abiogenesis singularity. And you know, these arguments can be constructed, as we've seen from Thompson, Lewontin, and Weiss, and this then renders universal common descent as an axiom, which appears sound, even elegant. The problem is it's not elegant, or it's not sound. It is logically incoherent. And this is not a matter of testing. You don't need to go out and look at the world. You don't need to put anything under a microscope. There's no observation required. You can see the problem from any armchair. If you formulate universal common descent as a deduction from these two premises, namely, one, abiogenesis happened from chemistry at a singularity. Two, only organisms cause other organisms. Then the theory rests on a contradiction. If premise one is true, premise two cannot be. If premise two is true, one cannot be. The contradiction is inescapable. And to my knowledge, it's never been resolved, because you're asserting two claims about the world which cannot be true at the same time. So universal common descent as a deductive inference fails long before any actual testing. The theory is wrecked on a logical shoal. And when I've discussed this with evolutionary biologists, they realize that this is a problem, and they say, well, look, whatever we think about universal common descent, even though we may regard it as the core proposition of biology, it's got to be testable, right? It can't be a first principle. So there must be traffic between theory and data that flows in both directions. This is philosophy of science, 101. Universal common descent must be testable against the evidence. It cannot be held as a first principle. It's got to be vulnerable to data should any challenges arise. So we'll take this as our starting point. We're not going to adopt the theory as an axiom. So let's look at some standard formulations. Here are three from textbooks that I used, or excuse me, the first two are textbooks. This is from a scientific article by Russell Doolittle in 1991. And notice the theory can be expressed very quickly, right? In a couple of sentences, there it is on the page. All organisms are related. All organisms are related to one another, and so forth. All life on Earth. Here's another pair, again, from textbooks that I used as a graduate student. Notice the modifier here, all. So we're making, again, that universal claim, all organisms on Earth. Now, notice something about the nouns here. They're generic: species, life, species, forms, ancestor, organisms, ancestor, descendant. These are very general descriptors. They don't refer to any particular species, any particular organism. They are nouns at the most abstract level that one could have in biology. Same as the case here. Organisms, 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 life, common ancestral cellular organism. This is very important. These terms, because of their general character, apply to any organism and to no organism in particular. All right, keep that in mind. Now let's look at the verbs. So here's my little PowerPoint collection. Okay, these are all organisms. Of course, they have many distinctive peculiarities representative of their species, but these terms apply to all of them in general and to none of them in particular. So universal common descent, in terms of its descriptive content, gains its universality by abstracting away from the specific features of living things to the most general. And this is true also for the process or mechanism terms in the textbook formulations. All right, so let's look at those. Here we have linked, related, descended, existed. This could be true. Linked could be true of a sexually reproducing organism, an asexually reproducing organism. There's nothing about that verb of action that has any biological specificity. What it tells you is A gives rise to B, again, at the most abstract level of formulation possible. It's true for these other textbook formulations: descended, diversified, produced, connected, and so forth. So the verbs of action that we see in these textbook expressions also describe abstract relations that are entirely general. Now, why is this a problem? It's a problem because while the perfect generality of universal common descent is very attractive, because scientists want theories that compress as much as possible into as little as possible, that's very attractive. In fact, you can express this theory as a single general proposition. This is often what you would find in a biology textbook, right? One sentence and you've got the basic concept there. That simplicity and generality is very powerful. It also, unfortunately, is impossible to test, because it doesn't tell you anything that you can go out and check, right? There's no observational or empirical content beyond the most general claim that there was an organism called Luca that existed in the past, and every living thing has descended from that entity. So we're right on the edge of an axiom, and we're not going to hold the theory as an axiom. We want to pull the theory away from this abstract generality towards testable content, if we're interested in testing it, okay? Now, that's our next question. How are we going to test this proposition? And here the problem takes a counterintuitive turn. So the best way to look at that counterintuitive turn is via a couple of thought experiments. So part two, how does universal common descent generate predictions? So we need a basic model of theory testing, and this is just about the most basic one available. You've got a theory, and it predicts or entails some observations. So you go to check, right? You go to see if those observations obtain. If they do, the theory is provisionally confirmed, not proved. Confirmed in the sense that it is at least consistent with what you observe. If they don't, it's potentially disconfirmed or falsified. So this is known as modus tollens. You learn this logical framework when you begin to take epistemology, philosophy of science. As I said, it's the most foundational or basic formulation of theory testing that one could have. Of course, actual theory testing is much messier than that, but we'll start with modus tollens anyway before we get into the weeds. So we want to know, what does universal common descent predict that we can observe? So let's try our first thought experiment. And the point of the thought experiment is to
You there?
Yeah, you know what? When I came back, I saw the mute that I was muted.
No, I was muted.
Oh, okay. I don't know. We lost our connection there briefly.
Okay, I don't know what happened. Well, you can keep going.
All right. Can you see that?
Yeah, I can see that. That's we were at the modus tollens. I think we might have been at the next slide. I'm not sure, but
Yeah. Okay. Well, you can edit out the little hitch there.
Or we'll leave it for fun.
For realism.
For realism.
All right.
The messiness of
there you go. Messiness on display.
Yeah.
So we want to know, do the observations confirm or disconfirm universal common descent? I'm sorry, my slides weren't advancing for a second there.
You're good. You're good. That's, I think I remember the
that's where
the universal Ghostbuster symbol, you know.
Yeah. So this ends up being very, very tricky. Surprisingly so, as you'll see. All right. So to do the thought experiment, we need a knowledge base. So here is a ribosome making its way down to messenger RNA strand. And I have to tell you, this kind of big marshmallow representation of the ribosome is what I had in my textbooks when I was a college and graduate student. You tend not to see these anymore because we know, in fact, as I'll discuss tomorrow, that the ribosome is an incredibly intricate factory with all kinds of bells and whistles. But what the artist is doing here is eliminating all of that so you can focus on key features. So I don't know if you can see my, whoops. I'm trying to visualize, I'm trying to get my cursor to come on here. I don't think it will. Anyway, we'll keep
That's
going.
all right.
All right, so you can see coming out of the tunnel in the large subunit is the nascent protein with its amino acid linkages.
Which on the first or the second or the third one, with the protein coming out, which one is that?
Oh, it is the first one.
The first one. Okay.
Yeah.
Gotcha.
So as the ribosome is going down the messenger RNA strand, which is specifying the assembly instructions for the protein, it comes to a stop codon in the A site. Now, there is no transfer RNA that recognizes a stop codon, because if there were, the process would continue. The process of protein assembly would continue. So what recognizes the stop, which tells the ribosome, all right, you've read the relevant information for protein assembly. What recognizes that is a protein called release factor. And as its name implies, it comes in and it hydrolyzes the ester bond there in the P site that keeps the nascent polypeptide, the nascent protein, connected to the ribosome. So release factor comes in at the A site, cuts the baby protein free, which then can go off and fold, and then the ribosome separates into the large and small subunits and goes off to find another messenger RNA strand to do the job again. Now, release factor is critical for this process. In other words, it's not a peripheral player. It's essential for protein assembly because it does that job. If you don't have release factor, what happens is, or if release factor is disabled in some way and doesn't recognize the stop, what happens is the ribosome will go down the messenger RNA strand, arrive at a stop. Because there's no transfer RNA to recognize the stop, the ribosome will just idle there. And the protein will remain attached to the ribosome and it won't fold. So you can see that release factor is critical for the process of protein assembly. And actually, ribosomes, I think, far more than some of the other molecular machines that intelligent design people are fond of, such as the bacterial flagellum. If I had to pick a molecular machine that was the sine qua non of the living state, and really it is, it's diagnostic of life on this planet. Viruses like COVID, SARS-CoV-2, have to hijack cells that have ribosomes to do this essential function of producing their proteins. Viruses, by definition, lack ribosomes. So it is the key player in the history of life, the story of life on this planet. And I'll talk about that more tomorrow. Anyway, bottom line, the key fact you need to take with you into the thought experiment: release factors are essential cellular hardware. Okay, so let's assume the truth of this geometry with Luca there and the three major domains descending from Luca. And we say, well, if this is correct, and if Luca possessed a DNA genome, what would likely be the case in terms of the hardware present in that cell? Well, as I said, it would have a DNA genome. It would have a genetic code that's mediating information transfer between the DNA and RNA and eventually protein assembly. It would have a ribosome. And as part of the ribosomal apparatus, it would have release factors. Okay. because they're demonstrably essential, we can say, well, we can probably reliably extrapolate out from that to the rest of the tree of life. In other words, if release factors are present in LUCA and performing that essential function, all of the all of the descendant groups that have LUCA as their ultimate ancestor would also have release factors, and they're likely to be homologous. So we can infer out from LUCA into the three domains and say, wherever we go in the tree of life, we're going to find shared release factors that descended from the minimal set present in LUCA. So we can do it this way. There's our prediction, conservation of release factors. We've got the three major domains. All right, that completes the setup of the thought experiment. Now, ask yourself, and I've actually done this with students, place your bets. Will the release factors of the three domains be homologous or not? And for homologous, you should take that to mean exhibit topologically congruent three-dimensional folds, so that these folds, if you went to the protein data bank and used their viewer, you could rotate them and they would be, in three dimensions, they would be congruent. So I have my two hands here. They're clearly, clearly hands, but they're not congruent, right? My thumbs are on opposite sides. So I've got a left hand and a right hand. These proteins would be topologically congruent in three dimensions, meaning you could superimpose the folds on each other. All right, prediction, they're going to be homologous. In fact, they're not. So here we see release factor on the left in bacteria and release factor, the crystal structure, in eukarya. The only thing that's the same is the GGQ motif that does the actual hydrolysis function. You cannot superimpose these folds. And again, if you're curious about that, go to Protein Data Bank, look them up, and just rotate them, right? These are definitely not the same proteins, even though they're doing the same job in the cell. All right, so our prediction is exactly wrong based on universal common descent. Now, when this was discovered, it had people scratching their heads, right? When the crystal structures for these proteins were solved, they turned out to be structurally distinct. And this was not at all what was expected on the basis of cell biology, knowledge of ribosomal function, and universal common descent. Something in that bundle of theory and assumption and data, something in that bundle went wrong because the prediction turned out to be exactly wrong. All right, no homology between the two. Now, you say, well, the prediction failed. Maybe we should rethink universal common descent. That didn't happen. It's very important to recognize what happens now in the logic tree when a prediction fails like this. You've got to go back into the logic tree and look to see what happened. So release factors are not homologous. That's curious, that's odd, but universal common descent, still okay. So if you go into the literature around the time that this was discovered and then people began thinking about it and publishing on it, you would see comments like this. The evolution of well-established release factors holds several unsolved puzzles. There's no strong evidence for an evolutionary relationship between bacterial release factors and their counterparts from archaea and eukaryotes. Ergo, it is unknown how termination was mediated, how that essential role was fulfilled in the last common ancestor. More recently, these commentators said, translation termination is essential, and it's universal across all Earth life. But the lack of universal conservation of its components raises questions about the importance of this step in the ancestral translation system. So these two authors speculate that translation may not have been essential at all once upon a time in the past. So falsified prediction, you go back in the logic tree, you can, throw a bunch of question marks in there, or you can say, as these two authors do, well, maybe translation didn't have to be terminated. Never mind how that could possibly be the case. That's one move that you can make when you go back and look at why the prediction failed. So it turns out universal common descent has some silent partners in the prediction business. When the predictions fail from universal common descent, this is not what happens. We can call this naive falsificationism. You don't go from failed prediction right back to throwing out universal common descent. This example and many others that I'll show you tomorrow show us that the situation is actually more complicated. Common descent is coupled with an auxiliary theory that enables us to make predictions, because common descent is perfectly general. It doesn't actually tell you anything about the world until you couple it with other parts of your biological knowledge. And then that lets you make predictions. So you got this complex bundle here. And what happens is when there's a failed prediction, typically the auxiliary theory takes the hit. And universal common descent is protected or insulated from empirical challenge. So an analogy that I've used in years past that I think is suggestive is a sort of the model of a medieval castle, right? Where the queen is in her throne room, deep in the center of the castle. And the barbarians are coming over the moat, and they're coming over the castle wall, and they're making their way into this palace, this castle. They never get to the queen. She's well protected in her throne room. And what gets slain, what dies by the edge of the sword, are the courtiers and the soldiers and the people in the outer layers of that castle, while she is protected. Now, most analyses of universal common descent would stop here. Of the testing of universal common descent would stop here. With this moral: take some wisdom from Pierre Duhem and from Quine. There's Duhem, there's Quine, who told us, and it's true, that when we confront our theories with the world, so we got our theory, we generate some predictions from our theory, we go to look at the data, what we bring to that task is always a complex bundle, never just a simple proposition. So when a prediction fails, it's often very hard to say why. Where was the problem in that complex bundle? Was it with theory A or theory B or some assumptions that we made, or maybe there's a problem with how we're interpreting the data and so forth? So a failed prediction does not prima facie tell you where the problem is in your complex bundle. That can be true, and I think it is true. I think the history of science shows that, but it doesn't solve the problem for universal common descent or for its testability. So remember our original prediction in the case of this thought experiment. We predicted homologous release factors. Okay, let's tweak our auxiliary theory. Okay, so we say, well, maybe translation termination wasn't essential in the past, or there's a range of variation in release factors that we failed to take account of. So we tweak our auxiliary theory in such a way that we can predict non-homologous release factors. Now the problem is we have covered the waterfront. Homologous and non-homologous release factors is all the possible data on these molecular actors. In effect, our theory, universal common descent, will be consistent with whatever we observe with respect to this particular function and the players that carry it out in cells. Maybe they'll be homologous, maybe they won't. Hard to say. We've tweaked our auxiliary theory in such a way that we can predict whatever we observe. That's a problem, because a theory that is consistent with whatever you observe is untestable. It's actually not telling you anything about the world, because whatever you find, you can find a way to harmonize it with your core theory. So first thought experiment is done. This is the no answer. So can universal common descent be tested? If you let the auxiliary theories take the hits consistently for failed predictions, even if you want to test universal common descent, you render it functionally an axiom, insulated from empirical challenge and beyond the reach of contrary observation. So let's look at an even more telling example, I think, because it's really throughout the biological literature from the mid-1960s, really until quite recently, and that's the prediction of a universal genetic code. So let's continue our no answer to our opening question. Quick summary of the main points so far. Universal common descent cannot be an axiom. It must be testable against data. Its perfect generality, however, requires helper or auxiliary theories to make testable predictions. This would not work. You don't want a bunch of question marks. You want something that you can actually go out and check. So in the case of the universal genetic code, it was thought, once the code was elucidated, that it could not vary. So let's call that the functional invariance of the code. So we couple universal common descent to an auxiliary theory about the functional constraints on the code, on the mapping that exists there between nucleic acid and amino acids and protein assembly. And that gives us a prediction that the code should be universal. So just to sort of motivate our intuitions or pump our intuitions, to borrow Dan Dennett's term a little bit more deeply, let's just arbitrarily assign some numerical values here. So we say universal common descent by itself, it's too general. Doesn't predict anything, or what it predicts is unknown, question marks or 0, just to give some arbitrary numerical values. All right, so let's couple it now to an auxiliary theory. And let's say we get a prediction of 7, right? Notice here where all the strength in the prediction is coming from. If universal common descent by itself tells you nothing, yet when you couple it to another theory, you get something that you can check. All the work in that prediction is being done by the auxiliary theory. And all universal common descent is providing you is a modifier, right? The term universal. You've got a logical modifier there, but the actual content is coming from the auxiliary theory. That's very significant, as you'll see, right? I remember when I saw this as it was a few years after I finished my dissertation, I wanted to kick myself, right? I'm looking at this and I realized all the work here is being done by the auxiliary theory in terms of what I can actually check. All the content is flowing from that auxiliary theory. And it struck me, somebody else must have noticed this, okay? So I went out in the literature, and sure enough, it was widespread. Among people who thought about this problem, the realization that universal common descent itself was untestable was widespread in the biological literature. So I'll give you a couple of examples. Lars Vogt, at the time this paper, he's a biologist who does systematics. The time this paper was published, he was at Harvard, and he was looking at the testability of cladograms. These are branching diagrams widely used in systematics to show the relationships of different groups of organisms, okay? And in this paper, notice the title, The Unfalsifiability of Cladograms, he says, any tree hypothesis is logically congruent with any distribution of characters along its terminals. So in a cladogram, all the species that we actually know or have evidence for occupy the ends of the branches, and the nodes further down are abstractions, like vertebrata or metazoa, right? A metazoan is an abstraction. It's a multicellular animal. Okay. He says when you look at these cladograms, they don't rule out any specific character state distribution. That is, you can't think of any observation just from a cladogram itself which would let you falsify that cladogram through modus tollens, which is the model of testing that we've been using, because there's no deductive link between the cladogram and what's present at the terminals. In other words, common descent by itself is untestable. Oliver Riopel, who's here in Chicago at the Field Museum, in 2008, he made the very same point. He said, there's no universal law of evolution that governs character distribution. So you can have any particular arrangement of characters on a cladogram, and that cladogram might still be true. It might still be the case, meaning testability is lost because you can't go from that back to falsifying the cladogram itself. So I was, astonished to realize that people who think about this problem have said, well, actually, universal common descent, strictly speaking, is untestable. Well, I then put together some examples for myself to try to work this out to see, well, how then do people try to test it? And suppose we have here a population of hypothetical eukaryotes, single-celled eukaryotes like yeast, and they have gene XYZ, okay? And we want to ask ourselves, what does universal common descent predict about gene XYZ? What should we expect to observe? Well, here are all the possible outcomes under a hypothesis of common descent. So we have the ancestral state, and the sort of orange box there represents the gene. That's a DNA strand, chromosomal strand. It's just a cartoon. We have the ancestral state, 5 positions there on the left. Well, you could have no change at all. Complete stasis. The gene is under very strict surveillance by natural selection. There's no change at all. That's one possible outcome. Or you could have partial changes. Some mutations modify the sequence, but detectable similarity remains between the descendant state and the ancestor. Or you could have a complete change where mutations saturate the sequence and eliminate all similarity. The gene is still there, but just by using a matching algorithm, you wouldn't be able to identify it. Or you could lose it entirely. Genes are lost. This gene is lost. It's eliminated from the chromosome, or it's replaced by horizontal transfer from another species. So these on the right are all possible descendant states starting from our ancestral state. Now, universal common descent, because of its generality, needs to be consistent with all of them, okay? Because what we're saying is there's some geometry of material descent where all those outcomes on the right could possibly have occurred. So the only way we're going to be able to make a prediction is by interposing a rule that comes not from universal common descent, but from some other part of biology. Because otherwise, universal common descent is going to be consistent with whatever we see. We don't want that to happen. That's untestable. So we need to borrow a rule from somewhere else, okay? There's a German proverb, sardonic proverb: if the rooster stands on the dung heap, it will rain tomorrow or it will not rain tomorrow. Well, we've got everything covered, right? We don't actually know anything about the world because we haven't ruled anything out. So to avoid our German rooster, right, situation, we've got to borrow a rule from elsewhere. And that rule, this is very important logically, it needs to exclude certain outcomes. They cannot occur. So this is what happened with the prediction of the universal genetic code. It was thought when the code was first elucidated that it could not vary, because the code affects every protein in an organism. Even a bacterial cell with just a few hundred proteins, the code has to apply to all of them. Ergo, if you change it, you will affect all of them. And that kind of deep functional constraint was thought to be something that could not vary for functional reasons, or you would lose the cell, all right? So here's our rule, functional invariance, and we put it here between Luca and the organisms that we observe today. And we say, if this rule obtains, we should expect to see the same code in any organism we examine today. So this is a prediction that you can find in the biological literature from the mid-1960s, when the code was elucidated, up until really quite recently. In fact, even before the code was elucidated, typically that is said to have occurred in 1966. In 1963, 3 years earlier, in an article in Science, Heingardner and Engelberg said, if there is a code, it would remain invariant once it came to be. Why? Because a mutation would change every protein in an organism. That can't happen. Therefore, once the code is established, it will remain invariant. So here's our three-domain representation of the tree of life again. And we say there's going to be a genetic code present in Luca. There it is in the RNA representation. This is the so-called canonical or standard code. And it is mediating, excuse me, information transfer from the DNA library, the realm of nucleic acid, to the realm of proteins, the cellular actors. Very important functionally for any cell. Okay, so we say if Luca had a DNA genome, which that's our most reasonable inference from everything that we observe today, it's going to have a genetic code, it's going to have proteins, that code is going to be fixed functionally. There's our prediction. Mutations were thought to be inevitably lethal given their likely effects. So we can extend that prediction then from Luca out into the three domains and say, wherever we examine an organism, we will find the same genetic code. So this existed as a prediction in my textbooks. I remember vividly learning it as an undergraduate studying evolutionary biology and genetics at the University of Pittsburgh in the early 1980s. And if you get a college textbook from that period, the early 80s, mid 80s, and go to the section in the textbook on why do we think all living things share a common ancestor, you will find this prediction there. Very clearly articulated and resting on the strong logic that we see here on this slide. Now, Dawkins in The Blind Watchmaker says it's a near conclusive proof. This is how he knows universal common descent is the case. In fact, I think if you go on YouTube, there's an interview, there are multiple interviews, in fact, with Dawkins where he says this, that the question is put to him, how do you know universal common descent is the case? He says, the genetic code is universal. And he says, I regard this as definitive. It's a near conclusive proof. So again, here's our little schema. Here's a typical expression. This is 1985, a textbook that I used. It would be retained, the code would be retained because any change in it would be disastrous. We expect the genetic code to be universal if all species have descended from a single ancestor. So there's our prediction, and we can place our bets. So 1963, the prediction is first made, and I have never found an earlier prediction than that, but it's interesting to me that 3 years before the code was actually solved, biologists knew there had to be one because of how protein assembly worked, and that you were going from one realm of chemistry, one alphabet, the 4 nucleobases of DNA, to the 20 amino acids at the time, now it's 22, the 20 amino acids of the alphabet in protein assembly. You had to have something mediating information transfer between those two kinds of chemistry. That prediction was already there in 63, 3 years before the code was actually solved. Okay, I think we should stop here. And we'll stop here. We'll pick up this thread tomorrow morning, and then we will look at several other examples of these kinds of predictions dealing with molecular data. And always the question to keep in mind is, if universal common descent were false, how would we know? Right? If we're making these predictions from the theory that are consistently crashing into observation, at what point do those failed predictions make their way back to the theory itself? What's happening? Is traffic really moving between theory and data and then going from data back to theory? Or is the situation more complicated? And tomorrow, I'll give several examples looking at the ribosome, looking at key enzymes involved in, for instance, modifying transfer RNA, basic molecular hardware that is not universally conserved the way that universal common descent predicted. We'll look at several, it's a theme. We could spend hours and hours on it. We'll just look at some striking examples, and then we will shift our focus to where I think the problem really lies, and it's in this puzzle that I'm calling Silber's paradox. So I'll stop there. We have a few minutes. If Jeff, if you have any questions.
Yeah, I think most of our questions will be tomorrow. So there's, just to clarify, I was kind of like thinking a bunch and listening. So, did you come to a conclusion? So there are different languages for each domain, or?
That is part of what I will say tomorrow.
Okay.
I don't want to get ahead of myself.
Okay, so we won't get ahead of ourselves. So is do the release factors, are they kind of the translator for that language?
Well, let me
do they like take the RNA message from the DNA and make it into a protein, right?
Well,
that's based on, and how they do it is the language. Would that be the best way to describe that?
Let me see if I can think of a good analogy.
I'm a computer person, so but there's plenty of people who don't have a scientific, who listen to this, and some who do.
Yeah. So. Maybe a good way to think about it would be comparing a 4-cylinder internal combustion engine. Mm-hmm. So you've got your cylinders where the combustion is actually going to occur. They're going to drive a piston, which will connect to a transmission and so forth. So have that architecture in mind, or a 6-cylinder where you've got literally a cylinder, right? That is going to be moving in and out in terms of the burning of fuel and oxygen via combustion. Compare that to a rotary Wankel, right? No cylinder. The cylinder is gone. You still have a space where combustion is occurring. The engine is still based on, a high energy fuel being combined with oxygen, but the geometry of the engine is totally
gotcha.
Okay, so in this case, if you look at these two release factors, can you see my mouse right now?
Yes, I can.
Okay, this GGQ motif, those are 3 amino acids right there.
Yeah, I see them.
Its corresponding active site, so to speak, is over here in this protein.
Okay.
But these are different. They're as different as a 4-cylinder engine is from a Wankel in terms of the overall geometry of the fold. So you can't superimpose them on each other. No, they're not topologically congruent. My 2 hands, no matter how hard I try, I cannot superimpose them on each other, right? The thumbs are always going to be on the opposite side.
Right. Yeah, I gotcha.
Okay. So they're doing the same job, right? There's a functional role. Hydrolyze the ester bond that's holding that baby protein to the last tRNA. All right. You've got to cut that protein free to go off and fold. That's the job they're doing. But when you look at the actor itself, it's not the same. So the question is, if that ribosome and all the parts involved in the ribosome were present in the last universal common ancestor, how did this fundamental difference arise, right? As you're diverging out from the last universal common ancestor into the three domains.
Right. So using a computer analogy, this would be like in the core kernel of the operating system. This is like a or a car analogy. This is the engine. You know, it's not like you're changing like a doorknob or something on a car.
Right. Think about the architecture of your laptop, or the architecture of, I have a iMac here, right?
Mm-hmm.
I take the back off, which Apple never wants you to do. I get inside and look at it, right? And it's not an Intel CPU. It's not an AMD CPU. It's not even a CPU at all. It's some other
a crystal or something.
Yeah, right.
Yeah.
So, any complex system will have elements that are more deeply entrenched in terms of its key functions than anything else. This iMac does not have to be silver and black. It could be magenta, right?
Right.
That can change. What can't change? The power supply. I've got to have a functional power supply in this artifact to do anything, right?
Right.
So when you go down into these core enzymes, these core functional players in cell biology, what you find is that they are remarkably not conserved. So tomorrow, to the degree that we can squeeze into an hour and 10 minutes or an hour and 15 minutes, I'm going to give you multiple examples of key centrally located players in cell biology that, contrary to universal common descent, that prediction of conservation, they're not conserved. And they're not conserved to a degree that's mind-blowing, right? So the impression that you would get from a cell biology textbook is that what's true of, in fact, Jacques Monod said this, what's true of E. coli is true of the elephant, right? Once you understand the core basics, you can take those and they'll be the case pretty much whatever system you look at. That isn't true. And the reason we discovered it isn't true comes from the technology of whole genome DNA sequencing. Okay. Because, really we could examine the parts on a wide scale and see the lack of conservation. So this is we got the logic on the table today. Tomorrow we'll look at a lot of examples.
Gotcha.
And then we'll look at Sober's paradox, which I think is really the heart of the problem.
Okay, so but to my question, this piece does the coding, like when we said the universal, you were talking about the invariance of the code, the language.
Actually, it doesn't do the code. Let me just show you this cartoon again.
Oh, so that one does. I got you.
So here's the ribosome. Moving along the messenger RNA strand.
And the release factor is just part of the ribosome.
It's part.
Yes, I got you.
Yeah, it's right there. And these are RNAs. They have an amino acid attached here at the CCA stem. This is a protein. Okay, so these are transfer RNAs in this position. This is a protein, and it's the molecular scissors that's going to cut this free.
Oh, gotcha. I gotcha. Okay.
Go off and fold. And so what it's recognizing here is when the stop codon in the instruction set is arrived at, release factor says, okay, I'm on deck. I'm on stage.
Right. So for our audience, the language is how to translate that UAG.
Right.
Or the CUA or something. Okay.
Right.
Since I know we're up against time, I guess the last thing, for the auxiliary theory, that's kind of like what you're saying, that's kind of like the fall guy or the patsy.
Well, you've got your core theory, universal common descent. You've got an observation that doesn't agree with prediction. Who's going to pay? And the history and philosophy of science has shown, in fact, Imre Lakatos developed a whole theory about this, that scientists tend to protect their core. right?
Mhm.
The core theory is relatively insulated from observational challenge because, after all, it is your core. You've got a lot of other things that depend on it. So when you have an observation that doesn't agree with a prediction made from the core, you make the courtiers and the soldiers pay the price, and you protect your queen. And
Right.
that's fine, right? That's actually rational. The problem is, if you do that enough, the core ceases to be testable and becomes an axiom.
Right. I mean, we all do that with our theories, and it's a healthy adjustment, right? But when
It's
you
a
do it,
healthy adjustment. We do that with life, right?
yeah.
Right? Like, I know my wife loves me. So when
But
she says,
the more that is an axiom, the more that's in theology, I'm a presuppositionalist. The more it's like we wouldn't do that with like the law of non-contradiction, right?
right,
But something further away from the law of non-contradiction, we would be more ready, make more adjustments.
right.
But the further, but yeah, as you say, like if you do that too much, then it just becomes like it becomes a mess, right? Yeah, I gotcha.
Yeah, you want the traffic, to use the two-way traffic metaphor. You want the traffic to eventually to be able to get back to the core.
Yes.
And to me, it's a time in biology that's incredibly interesting, because, as I'll show tomorrow morning, many biologists, for reasons that have nothing to do with intelligent design, are now saying, I don't buy the single tree. The single tree picture has failed in the light of what we've learned over the past 25 years. And what they're doing is they're sorting that complex bundle, and they're saying, I actually know more about the auxiliary theory than I do about universal common descent. Universal common descent has got to go. The queen has got to be deposed. So that's something we'll pick up tomorrow morning, and we'll look at a lot of molecular examples. And then I think I can squeeze this all into an hour tomorrow. And then Sober's paradox.
Okay. Well, thanks for coming, and Lord willing, I'll see you tomorrow. And
yeah, so it's 9 a.m. your time, 8 a.m.
I'm going to stop recording here.